Hi everyone, the first practical that we're going to do is on pipetting. I just want to run through some brief lab safety information first of all. So it is extremely important that you always wear a lab coat and gloves during the laboratory, that you know the location of the fire extinguishers and the fire exits, as well as the eye wash stations and the first aid kit. These will be pointed out to you at the beginning of the lab. You're never to eat, drink or smoke inside in the lab, or never use your mouth to pipette any liquids. Always use the pipette fillers that are provided. It's also important that you're aware where the correct waste bins are for particular types of waste. Again, these will be mentioned to you during specific practicals. Glass pipettes or sharps should be disposed of in the sharps bin, or any kind of breakages such as glass. You should inform your demonstrator or the lecturer in the practical. It's also really important that you clean and wash up after yourself. For the lab write-up, you're to provide some minimum pieces of information at the beginning, such as the title and the objective. You don't need to provide the materials and methods in your lab copy. You should only include detail that's different from the manual. The main section that you should be writing about are the results and finally the discussion. So the results will include things like your printout, any graphs that you generate, and any tables as well that you fill in. There should also be a brief discussion and conclusion, and you should also answer any questions that may be asked in the lab manual. Copies are marked on the objectives and the results and the discussion, as well as any questions as well. So today's lab is mainly about pipetting, and in particular about precision and accuracy. So it's important, I suppose, to understand the difference between the two. Accuracy is essentially how close you are to the true value, so accurate measurements give the true value, whereas precise measurements give the same value repeatedly. So if you were to repeat, in this case pipetting, at the same volume, and you were to pipette inaccurately but precisely, that would mean that you were good at pipetting the same volume, but that same volume was incorrect. It wasn't the true value. What we want you to be able to do is to be able to pipette both accurately and precisely. So therefore, pipette the true value, but also pipette the true value several times, the same over and over again. So you'll be asked in this practical to calculate precision and accuracy. So to calculate these of a group of measurements, you need to know two details. First of these is the mean, or the average, and also the standard deviation. So the mean is the sum of a set of numbers divided by the amount of numbers. So for example, if I was to ask you to pipette a volume of something 5 times, the mean of that would be those 5 different measurements added together and then divided by the number 5. The variance then is the distance that each of these measurements has from the mean. So having got the mean, you can now calculate the variance, which is simply just looking at how much each of those individual values, those 5 values, varies or differs from the mean value. So from that then we can calculate the standard deviation, which is the square root of the variance. And there's a formula for that, but you will be more likely using your calculator for this. So just to briefly introduce you to pipettes, pipettes are devices that allow us to deliver very, very small volumes, and shown in the diagram here are 2 particular pipettes, a P1000 and a P100. It's important to note that these pipettes have different volume limits on them. So the P1000 can measure volumes accurately from 200 microliters to 100 microliters, whereas a P100 is recommended for the range of 10 microliters to 100 microliters. Each of these pipettes has a different colored top, and this colored top usually corresponds to the same color pipette tip, which is put onto the end of the pipette. In terms of using them, you should watch the video provided on Blackboard for the demonstration, but I suppose on this slide here it just shows you up close what the dials show. So the dial actually shows you the volume there. In this case for a P200, it's pipetting 200 microliters, so you can see the 2 a 0, 0, and then a line, and then a 0 again. So this means it's 200.0 microliters. So you set the volume by turning the knob at the top of the pipette until the desired volume then is shown in the window. You should always make sure then before you actually use it that there is a tip, so the correct color tip as well, on the pipette before placing it in the liquid. And never try to use or adjust a pipette outside its volume range. So don't try and pipette 210 microliters with a P200 pipette. There's a specific way of using it, and again, it's best if you watch this for a visual demonstration on the video, but it involves depressing the button slowly, so the top button, to the first stop with the pipette tip on and place it into the liquid. You then slowly release the button to allow the liquid to slowly come up, and again try and avoid air bubbles. If you do notice air bubbles inside it, please start again, expel any liquid and start again. So slowly release the button to allow the liquid into the tip. Remove the pipette carefully and place the tip then where you want to displace the liquid. So if you're transferring it from one beaker into another, you'd remove the pipette from the first beaker, transfer the pipette then to the second beaker and place the tip where you want it to displace, and just depress the button slowly to the first stop again. You then wait a second and then you depress the button, the top button then to the stop, the second stop. So important for you to get a good feel for the pipette and to notice that there are 2 stops involved. So for this experiment, there's going to be 2 methods. Okay, the first of these is going to involve pipetting small volumes of liquid. So these volumes are indicated in your lab manual. Of a small coloured liquid, it's just some food dye, into a microtiter plate, a 96 well plate, using these pipettes. You'll then measure the absorbance at 450 nanometers. These are already preset for you on the spectrophotometer. You'll get a printout, and then from the printout, you should be able to calculate the mean, the standard deviation, and the percentage variance. So just a reminder then, the mean is just the average. So again, if you have 5 values, you add up the 5 individual values and divide by 5. You'll be able to calculate the standard deviation from your calculator. And again, please familiarize yourself with the calculator functions. And then for the percentage variance, it's simply the standard deviation divided by the mean multiplied by 100 for each of these. In the second method then, it's going to be involve the calibration of the pipette by weighing using a weighing balance. So what this means is that you're going to pipette different volumes of water into a weigh boat, record the weight, and then repeat it until you have 10 weights for that given volume. So you're going to have to dispense a number of different volumes for that. And again, don't forget to zero the weighing balance with the weigh boat by itself, because that actually has a weight by itself. So make sure it's at 0 before you dispense the water into the weigh boat. You then calculate the mean, the standard deviation, and the percentage variance as before.